Good afternoon, everybody. My name is Louisa Sylvia. I'm the director of the Office of Women's Careers at the Center for Faculty Development. It is my distinct pleasure to welcome Zohreh Amoozgar and Esther Warnert as our speakers for today for the Anne Klibanski Visiting Lecture. So I hope this isn't your first Anne Klibanski Visiting lecture. If it is, we'll give you a little bit of background anyway, because we want you to know why we're doing this wonderful series. This lecture series really got off the ground because, as you may be aware, the pandemic disproportionately affects women with the other responsibilities that they have, makes it more difficult for them to travel, do talks, network, collaborate. And as a result, we thought we could do this award to bring women together, faculty, clinicians, educators, research, and postdocs that have exceptional promise as leaders in their field who benefit from working together and speaking nationally and internationally together and mentoring and networking opportunities. So we have 36 faculty clinicians who have been awarded the Anne Klibanski Visiting Scholar Award. And I believe we'll show those in a minute. But first, if you don't know Anne Klibanski, she's the president and CEO of Mass General Brigham. She was actually the former director of the CFD. And really got this group off and running. She's been extremely successful, both as a clinician and as a researcher. She's been a strong support and advocate for women over the years. And so therefore, it was very well suited that this Scholar Award would be named after her. Notably, she also established the Claflin Awards, which is another very popular award that really helps to support our faculty here at Mass General. And as I mentioned, these are the Anne Klibanski Visiting Scholars for this year. I hope you recognize one or two folks on this page. It's really an outstanding group, and we're very thrilled to have these women leading the way at Mass General for us. And these are some of our partners who have been partnering on the networking, collaborating side to support our scholars and as well to do these lectures together. Today, we're going to highlight Erasmus University because our speaker, Esther, is from Erasmus from the Netherlands, which is wonderful to have her here. Again, the beauty of Zoom is we can bring her from the Netherlands to your office or home instantaneously. And so, again, just a bit more about the lecture series. Please make a note of these. We put them in our newsletter every week. We really want people to come support our wonderful faculty and also hear wonderful speakers and hear some of the latest and greatest information out there across medicine and across the hospital. And importantly, these lectures are open to really everyone at Mass General as well as the host institution. So I hope that we have folks on from the Netherlands today. So without further ado, let me introduce our speakers. Again, Zohreh Amoozgar is a research fellow in radiation oncology. She is a PhD in pharmaceutical sciences from Purdue University in India. And she seeks to find ways to alleviate resistance to therapy based on a specific microenvironment of tumors. So she's going to give a talk on reprogramming the tumor microenvironment to improve immunotherapy. Wonderful to have her here. And our second speaker, I'm going to go ahead and introduce as well, because they're going to go from one talk to the next, but we hope to have some minutes at the end for questions, is Esther Warnert, who's an assistant professor of radiology and nuclear medicine at Erasmus. And Esther really aims to build the bridge between developmental and clinical applications of advanced MRI techniques to assess cerebral physiology in both health and disease. And so she's the PI of the research line bench to bedside imaging biomarkers, which is wonderful. And she, as I said, is really going to speak a bit about going beyond the structure, advancing the use of physiological MRI in glioma imaging diagnostics. Hopefully I said that right. So thank you both for being here. I will stop sharing my screen and move on to letting Zohreh take over.
Well, hello everybody, and thank you very much for this honor and privilege to share a talk with my newly found friend and collaborator, Dr. Esther Warnert, through Erasmus program. So we're gonna talk together about Theranostics in glioma. And as an outline, what we're gonna talk about collectively and mostly our both work focused on microenvironment of brain tumors. We're gonna talk about elements of the tumor microenvironment, and I specifically highlight how every element in the tumor microenvironment of specifically glioblastoma which is the deadliest of all, is abnormal. Mechanism of resistance to therapy in glioblastoma that are multifactorial. I'm going to talk about 2 of them. And with help of Esther, we talk more about physiological MRI that we need to understand the tumor microenvironment in vivo and eventually advance this benefit to patients in the clinic. So my talk highly revolves around glioblastoma, which is the universally lethal primary brain tumor. We have made extensive advances in cancer therapy. However, these benefits do not translate to a primary brain tumor in brain that from the time we diagnose it until the time that patient unfortunately expires is roughly about 20 months with every new technique we have in our arsenal. And that begs the question, why and how we can improve this treatment. Knowing why can help us to understand how we can advance this therapy. So looking at the contribution of Steele Laboratory that I am a member of to understand the tumor microenvironment, we can think of tumor as very dysfunctional organs. They have elements of blood vessel, lymphatic vessel, matrix, cancer cell, and host cell. But I like to stress and emphasize that all elements exist, but they are all dysfunctional. And among them, I want to emphasize more about tumor blood vessels, that their impairment in function impairs the blood flow, delivery of molecules that we send into the body as therapies, as well as delivery of immune cells and the cells that are supposed to provide us therapeutic benefit. This environment creates hypoxia and low pH. And it's very important to stress that this environment is actually not homogeneous. It is heterogeneous all across the tumor. As here you can clearly see the hypoxia images that in various stages of tumor, you can understand and see how different it is. Looking and thinking about lymphatic, if you think of a normal tail of a mouse, for example, how clear you can see it in the left side of the picture, how you can see the architecture. And when you have a tumor growing, this is completely impaired. So you can think how these 2 elements, which is the blood vessels that are supposed to provide nutrients and the lymph that's supposed to organize and also make sure that we have a balance of interstitial fluid, both are impaired. Consequently, if we look at the interstitial fluid pressure in human tumors, what we can roughly think about as edema that can be caused, we can see, unlike the normal and healthy organs, in tumors, pretty much all of them are disturbed and that they have higher interstitial fluid pressure. When it comes to glioblastoma specifically, when we think about it, if we think of other 2 organs that can expand, we have brain that is inside the skull, so it doesn't have a lot of room to expand. Imagine if you have something that keeps expanding in it, and that's part of the reason why majority of patients that come to the clinic with glioblastoma manifest strong headaches, and they have a lot of difficulties there, causing them morbidity and mortality. So the consequence of a problem in blood vessels, as I mentioned before, is hypoxia and low pH, this pressure fuel, the pressure that we observe, it fuels many, many malfunctions, including immunosuppression, resistant to various forms of therapy, radiotherapy, chemotherapy, immunotherapy. It switches the metabolism of cells from aerobic to anaerobic, which actually causes a strong disturbance in the ability and response to therapy. So all of these are pretty much consequence of one abnormal element, which is the blood vessels. And we shouldn't be surprised why this is so important, because when you think about blood vessels, similar to immune cells or other things we discussed, they are pretty much everywhere. So locally, looking at the tumor, it generates and it produces factors that are causing a very strong immune suppression. It generates these cytokines that are causing increased amount of regulatory T cells that are known to be immune suppressors. It switches and shifts the phenotype of macrophages to be pro-tumor, supporting the tumor. It dampens the ability of dendritic cells to present antigen such that the T cell can recognize and kill tumor cell, and also significantly affects the endothelial cells on the walls of the blood vessels and microvasculature. And as a consequence, we have a localized immune disturbance, a localized, strong immunosuppressive environment. But it doesn't limit there. We have also a systemic immune disturbance as a consequence of what tumor produces and releases in the blood. Tumors produce vascular endothelial growth factors, TGF-beta, and prostaglandins, and consequently, we have increased amount of regulatory T cells. So even if systemically the body wants a response, it does block those responses. We have reduced amount of antigen presentation also systemically, and the outcome will be reduced amount of effector T cells that can respond to the tumor. So if we think of normalizing the structure and function of tumor vessels by because we have anti-angiogenic factors that block this new vessel formation, and we have pro-angiogenic factors that promote this vascular formation, which in this case are malformed. If we normalize this balance, we can have a normalized ability to produce blood in a correct way to the organ, and the outcome of this normalized function of blood vessels will be increased perfusion, increased oxygenation. We have reduced this edema that I mentioned to you before is deadly actually in case of glioblastoma. We have higher drug delivery, so the drugs can get to the tumor and kill tumor cells. And also it can be a major immune regulator. So these combination can be immunosupportive, favorable to the therapeutic outcome. And how this has been done in clinic, one of the salvage therapies for glioblastoma is use of anti-vascular endothelial growth factor. By normalizing this GBM environment, for example, here, you can see in the patient, the white matter that previously wasn't observable can be seen again in the patient. And having said that, again, as one of the treatment, it's gonna be work heterogeneously in the patient. It doesn't mean all patients are gonna benefit from this therapeutic outcome. For example, newly diagnosed patients, some of them have a better perfusion, some would not respond. And as it is observed, patients that have increased perfusion in response to bevacizumab or anti-vascular endothelial growth factor would live longer. But knowing this, we know that if you're gonna pile up these data together, you will lose the benefit because some respond, some do not respond. This actually makes the imaging MRI techniques and this type of interventions that Esther is gonna talk to us more about very important in patients to understand who is the responder, who is not the responder. And from our end, our laboratory and the Department of Radiation Oncology is heavily invested also in imaging, but I'm gonna talk about the other side, what we are doing in the lab partly. Again, still lab is quite known for imaging techniques, but I'm gonna talk only a bit about resistance to these therapies. So we do know circulating angiopoietin-2, and we discovered that as an indicator of therapeutic response. We find that the tumors that are treated with vascular endothelial growth factor at some point gonna resist this therapy. And what we find both in human and both in mice, we see that there is expression of angiopoietin-2 in the blood vessels, making them a potential resistant mechanism. Collaborating with other institutions, and here in a large collaboration with industry, with Roche, we used their antibody that can recognize in a bispecific manner both VEGF and angiopoietin-2 and antagonize them. So using that antibody, we went and checked in mice models whether we have a normal vessels as opposed to its standard condition where the GBM progresses versus when they are treated with bevacizumab or here B20, the mice version. And indeed, we see by analysis of vessels in terms of architecture, we have achieved reducing these abnormal vessels, and also we have a precise coverage that indicates vascular maturity. Moving forward, we have tested them in terms of their ability to increase survival benefit. In the left side, you can see a mouse model, GL261, which is a model that is heavily studied in GBM studies. And then in the right, you will see MGG8, a xenograft model that has been identified and made at Mass General. In both of the cases, compared to monotherapies of each, combination of blocking VEGF and angiopoietin-2 collectively increase survival benefit. And that made us ask this question that, why is that? Specifically because the MGG8 does have a normal vessel. Again, I emphasize, similar to the patient outcome, that things are not going to be homogeneous. There are going to be some patients who do have normal vessel. So we wanted to further interrogate what is the rationale and what is the reason. A major element of GBM is stroma, which is composed of heavily macrophages. So we saw with this therapy, the amount of macrophage and microglia is not changed. However, under this therapeutic condition, there was a phenotypical switch where the macrophages switched to be more M1-like or anti-tumor phenotype. And then based on this finding, if I were to summarize it very quickly, if we normalize the structure and function of vessel, we are not only affecting the vessel, but also we are affecting the tumor immune microenvironment toward tumor cell elimination. And right now, in the era of immunotherapy, in particular, immune checkpoint blockers that is standing as a part of standard of care in many ways, we want to extend this therapy that we know has failed in trials for GBM. We want to extend its benefit to the GBM patient if we can. So we looked how these glioblastoma tumors can be categorized. In general, we have three sort of tumors in terms of their tumor immune microenvironment. Tumors that are inflamed, they have a lot of CD8 in them. Tumors that don't have many, and tumors that CD8 T cells are sequestered around the periphery. And indeed, GBM is one of those that we have CD8, but they cannot reach. In these tumors, angiogenesis, which is this major vascular abnormality, is predominant. There is a large amount of MDSC, and I talked to you about with a strong prevalence of macrophages. And there is a reactive stroma that blocks this function of these T cells. So knowing this, we want to combine this material, which is blocking both VEGF and angiopoietin-2, and reactivating CD8 T cells in a way that we have more function at the end. And this has precedence because right now anti-angiogenic therapies plus immunotherapies are heavily studied in preclinical studies as well as clinical studies. And then moreover, what was interesting and important in our scenario is that we have seen in collaboration that angiopoietin-2 is a biomarker of lack of response to immunotherapy that provides us more rationale for this combination therapy. So moving forward, and in animal studies, when we block angiopoietin-2 and VEGF, which means we normalize vasculature in an extended time, and combine that with anti-PD-1 therapy, we were able to achieve a fraction of mice having durable response. And by durable response, I mean if we go back and re-challenge them with a new tumor, again in another side of the brain orthotopically, the tumor does not come back. So this question makes us further investigate how we can increase this impact of immunotherapy further. What can we do? And along the line, as I mentioned before, in the construction of tumor microenvironment is composed of many of these elements, including the immune cell. So we asked, are there other reasons for resistance, for example, to immunotherapy? And indeed, what we find is that one of the potential is by upregulating other checkpoints. So if we treat with anti-PD-1, we saw that CD8 T cells start upregulating TIGIT. And that was indeed both in CD8 cells, T regulatory cells, and CD4 T cells. And why is that important is because this TIGIT signaling is function differently. If it is high in T regulatory cells, it means it produces cells that can block CD8 function. If it also increases in CD8, it means that these CD8 cells are probably more dysfunctional. So with that in mind, we asked, is that a reasonable or attractive target in immunotherapy? And indeed, because it's overexpressed after therapy, because it's overexpressed, Tregs mark their suppressive phenotype, and because its ligand is also overexpressed in tumor cell, it made us to question that particular aspect. And actually, in collaboration with Brigham and Women, which is our sister hospital, we find that if we co-inhibit TIGIT and PD-1, we indeed can increase the survival benefit significantly. And also, if we re-challenge the other side of the brain, again, we will not have a tumor growing back in the fraction that did respond. So our future plan and outstanding questions are, how can we combine these therapies with the standard of care? Because as we know, standard of care is well established, benefiting patient, and new therapies should combine with the standard of care. And can we identify targets that can predict this response? And can in future, we can replace these systemic therapies with potentially localized therapies, so we can reduce this potential of resistance, also side effect. So in my conclusion, I like to highlight our treatment has to be tailored to the type of tumor microenvironment of a specific tumors. And with that, I like to thank Dr. Jane, who is the head of Still Laboratory, all members of Still Laboratory, who I had the pleasure to work with, our collaborator immunologist, Dr. Kim, and two important contributors to my progression in terms of, and assigned by Office of Faculty Development, Dr. Galit Alter and Dr. Bahus Panos, who made tremendous effort in introducing me to the community and promoting the line of research we are doing at MGH. With that, I thank you, and I will stop my share such that my colleague and friend Esther can start the presentation. Thank you.
Wow, that's great. Thank you, Zohreh. I'm gonna set up sharing my screen. So I think you should now be able to see my screen with the slide share. I see nods. Yeah, perfect. Wow, so thank you, Zohreh. So now I'm going to do my best to follow on in your footsteps. First of all, I'm very happy and grateful. So I'd like to thank the organizers once again for having us and inviting us to present our work. I am indeed going to focus on going beyond structure, advancing physiological MRI and glioma diagnostics. This is the background. So I work at the Erasmus Medical Center. You see this here in the background as a part of the skyline of Rotterdam, which is in the Netherlands. All right. There we go. To set the stage a little bit in terms of numbers, each year in the Netherlands, there are about 1,200 patients with newly diagnosed glioma per year. And I've looked up the numbers in the US, and of course, they're, you're a little bit bigger. It also means that you have about 83,000 newly diagnosed glioma patients each year. What this infographic shows is that if you then look at primary brain and CNS tumors, you see that glioma and glioblastoma make up of about 25% of these brain tumors. Zohreh was talking a lot about GBMs or glioblastomas, and as you can see here on the right-hand side, what they present, how they present often is with enhancement, and that is because, the vasculature, again, Zohreh highlighted, is poor. If you inject a contrast agent, so in this particular case, a gadolinium-based contrast agent, you see that the contrast agent will leak out of the vasculature, and that results in hyperintense signal here within the tumor. I also want to focus a little bit on non-enhancing glioma, just because I'm going to show some results of these type of brain tumors as well. You can see them here on the left-hand side. What this means is that this is a type of brain tumor that has an intact blood-brain barrier. And what you can see is that if you inject contrast agents, As you can see in the T1 post-contrast image, you do see that contrast agent is there. You see the hyperintense signal in the vasculature, but this actually doesn't leak into the tumor tissue. Now, although originally or classically, these non-enhancing tumors were often deemed to be low grade 2 to 3, we now know with the updates in the classification of brain tumors in 2016, that actually, even if you look at these non-enhancing gliomas and you take specific molecular parameters into account, you can see already that with the blue line here, there are certain non-enhancing gliomas that behave almost a very similar behavior as GBM with an even with a similar dismal prognosis. And just to set the scene a little in terms of what type of images and tumors we're looking at here. Then setting the stage for standard MRI. So here I'm highlighting the different scans that we would do, and I'm highlighting here as well that this is in the Dutch national guidelines. We do mainly structural scans. We do T1 weighted scans before and after contrast injection, and we do T2 weighted scans. It means that if you go to a hospital in the Netherlands, basically you get these scans made if no additional scans are asked for. We then see the addition of the diffusion weighted imaging, the apparent diffusion coefficient on the right-hand side. This is what is now the consensus brain tumor protocol for clinical trials, as has been published in 2015 by Ellingson and al. Now I'm going to talk about physiological MRI, and I'm already highlighting the word advanced here a little, or putting it in between brackets, because maybe most of you will recognize all the way on the left, a relative cerebral blood volume. This is a dynamic susceptibility contrast scan, which is actually quite often already acquired in the clinic as well. It's a measurement related to perfusion that you can also assess with arterial spin labeling. So that's the cerebral blood flow there. I'm also going to discuss vessel size imaging, oxygen extraction fraction, and amide proton transfer imaging in the coming 20 minutes. I'm also fully aware that this is not the full exhaustive list of all the advanced scans that you could potentially do, but these are just the scans that are going to pass by in the coming 20 minutes. Now, why would we want to do physiological MRI? And here, again, I would like to thank Zohreh for enlightening me why you would want to measure the tumor microenvironment. Well, we've heard just now, it's so important to probe the tumor microenvironment. We know that the tumor, the behavior of the tumor, but also whether or not it's resistant to treatment, how it will function or how it will respond to treatment will all depend on the tumor microenvironment. And in that way, it's ever more important to be able to assess the microenvironment in the tumor when it's still inside the brain. I'm highlighting here work by Stadelbauer and colleagues who recently made this move, and you can quite nicely see that if you use advanced imaging techniques, you actually can start to probe the tumor microenvironment within the tumors here as well. And this is really the aim that we're striving here for as well. Knowing that and being able to probe the microenvironment, Zohreh already mentioned, it might be very beneficial to look at, for instance, patient selection for treatments and trials. But I think the most important bit to highlight here is that by doing this, you will be able to address clinically relevant questions that currently structural MRI simply is not answering. So that indeed, again, patient selection, it could also help in more accurate diagnosis, having image-guided biopsies. And I think this list of potential topics will only grow longer if we advance the imaging techniques towards clinical application. So why then am I working this hard to advance these techniques? So I think an important aspect to highlight once again of these advanced imaging techniques is that they are emerging techniques. Most of them are relatively new. And that means that we're still really working on, well, how we should actually measure these imaging techniques well. So there is still a bit of development that is required in order to move these techniques forward. Then if we are happy with the acquisition, or if we do think, well, now we are set and we're certain that we can scan the same thing every time, we actually do need to validate what is measured. Most of these imaging techniques, they are based on physiological models, in particular, quite often made for a healthy tissue. And of course, when there's pathology present, you just want to make sure that what you think you're measuring is also actually reflective of what you're measuring. And of course, that's a very important aspect as well before translating any new imaging technique into clinical practice and trials. And as mentioned already before, I became a PI of my own research line, which is called Bench to Bedside Imaging Biomarkers, in which I really strive to generate an environment like this that covers all three of these topics here at the Erasmus Medical Center. In the interest of time, however, I would like to present the ITEM study to you. And this is a study that we're running currently at the hospital and that we have been setting up for the past year. And we started patient recruitment only last summer. So the ITEM study stands for Imaging for Tumor Environment Mapping, how appropriate. And what we do here is we aim to validate advanced physiological MRI in glioma. Now, what that means is that we do an MRI scan pre-surgically. So this is the scan the day before the patient undergoes surgery. In the surgery, we'll take biopsies that are taken based on the advanced imaging techniques. And then in the lab, we will look at biomarkers to see whether the pathology or whether the parameters that we're interested in with the advanced physiological scans are actually present within the tissue. So the ITEM study actually is built around two different projects. One of them is called Food for Thought, and the other is Making the Invisible Visible. And I very briefly want to go through both of these projects now, just to give you an idea about what we're doing. So again, the background of the Food for Thought project focused on oxygenation measurements. Well, I guess I don't have to stress once again how important it is that we can assess tumor hypoxia. We know that it stimulates poor neovascularization and tumor growth. It can diminish effects of therapy. As a slight plus, I think what is interesting to note already that Zohreh was also hinting at, we could potentially target this with treatment, but most importantly for me, we can also image this. So it's really important to bring these imaging techniques towards the clinical application, which is why within this Food for Thought strand, we're aiming to develop and validate a clinically applicable framework to assess oxygen delivery to brain tumors. It's centered around this very schematic idea of tumor tissue or brain tissue in which oxygen-rich blood will flow in, bringing in oxygen to the tissue. We can assess this with cerebral blood flow measurements with arterial spin labeling. It's then important to probe the local microvasculature, microvasculature. We do this with vessel size imaging with a hybrid EPI scan. So I will go into a little bit more detail into this image acquisition, type of image acquisition in a minute. And then we can also assess the oxygen extraction fraction, and we do that with a quantitative POLT technique. Now, what we have been doing is bringing this all together into an image acquisition protocol that we can do within less than 15 minutes in addition to the scan protocol. And we currently are working on finalizing the processing pipelines to make sure that we can really bring all of this information together to generate that tumor microenvironment map. This is work that has been done, that is currently being done by Fatima Razan-Farouche, who is one of my PhD candidates. And I just want to highlight some of her work and explaining the vessel size imaging technique here in the coming few slides. So vessel size imaging really starts from perfusion-based imaging that you can do with DSC. DSC, what you usually do is you do a gradient echo acquisition. You do this over a number of repetition times. You then inject a contrast agent. And as you see the contrast agent passing through the vasculature, you see signal change. And in general, this signal change is more or less proportional to the amount of blood volume in the voxel. Now with vessel size imaging, and we do this with a hybrid EPI scan, we can not only do a gradient echo acquisition, but at the same time, we also acquire a spin echo acquisition. The win of this is that the gradient and spin echo signals, if a contrast agent passes through the vasculature, they behave in a slightly different manner, and their sensitivities are slightly different towards different areas of the microvasculature. And the win of that is that if you combine both of these signal changes, you can then actually probe the microvasculature and get, for instance, a parameter of the mean vessel size that is present within a voxel. Now, what you usually see in this in healthy tissue, so if we look at this green line here on the left, is that you see that this DSC signal, you have a base, then you see the dip, and the tail usually matches more or less with the baseline. In a brain tumor where there's contrast agent leaking out of the tissue, this contrast agent leakage is actually affecting what happens to the tail of your signal, because it will constantly affect your MRI signal. And that is actually, of course, detrimental to the estimation of the parameter of interest, in this case, your vessel size imaging. So what Fatima has done, she's built on her very, very fresh off the press work. It only got published last week, where she was investigating different leakage correction algorithms for DSC imaging. She is now extending that to look at what is the effect of applying leakage correction in vessel size imaging. For that, she is looking at the results of the analysis of 12 glioma patients that have been acquired previously at the Erasmus Medical Center. And within these scans, or within these patients, we have done the hybrid EPI, or the vessel size imaging acquisition. Interestingly, what you see here on this particular slide, she's looked at different regions within the brain, so healthy tissue, normal appearing white matter and gray matter, as well as a non-enhancing tumor data as contrast-enhanced tumor area. And it's very striking. Immediately you see that, of course, it is important to include leakage correction when you assess vessel size imaging in the contrast enhancement area. So that's indicated here in the left top figure. An interesting finding here, albeit very small, she found significant changes in the normal appearing white matter, gray matter, and non-enhancing tumor here as well. Differences are very small, and this is, of course, only a small subset. She's presenting this work at the upcoming ISMRM meeting as well. So this is a virtual meeting. So if you're attending, try and find her presentation. However, what this means, or whether or not this is a true effect, is, of course, something that we can investigate within the item study. So it is, again, aimed at validation of these imaging techniques. We will aim to include 20 patients for this particular strand that will undergo vessel size imaging or oxygen extraction fraction mapping. Stereotactic biopsies will be taken based on these imaging, the new maps, in collaboration with the neurosurgery department. And we will collaborate with the pathology department to then validate these biopsies with immunohistochemistry stainings of hypoxia and microvessel density. So that's one strand of the item study, very much focused on the vasculature and oxygenation. Secondly, we have a strand that focus on making the invisible visible, that is focusing on APT-CEST, or amide proton transfer CEST. I wanted to very, very briefly introduce you to the chemical exchange saturation transfer imaging technique. Usually with the MR, what you can see here is that we're sensitive to the large free water pool. But what we do here is basically sensitize this signal to protons that are bound to proteins that are dissolved within this free water pool. You see these blue H's on the left, and they exchange with the free water. This will allow us to actually probe these signals within in vivo. And if this is a very nice review paper that shows how this technique works, but I always very much like this example to indicate, okay, how does this technique then actually work? So here they've put in a fresh egg in the scanner. You see the egg has egg yolk and it has egg white. And with this, we can probe protein content. So if you then look at the bottom line, we see in red that there is a high signal, in particular in the right below corner, high signal in the egg white. So yes, it does mean that if you're looking for a protein diet, don't eat the egg yolk. Stick with the egg white. Now, why is that important for imaging in glioma? Now, that's because recently it's been shown that APT signal, or that being able to be sensitive to protein buildup in glioma has been linked to being able to assess cell proliferation. This study here quite nicely shows that if you have high APT-CEST signal, they've done this in 34 patients, if I'm not mistaken, this really also matches with high cell proliferation, as they've assessed this with the Ki-67 labeling. And in result, that means that in this particular work, they also indicate that you can then use APT-CEST for glioma grading. But this is also the foundation in which the work focuses on using APT-CEST to differentiate tumor progression from treatment response and necrosis. And this is, of course, an important aspect in the clinical management of patients after they've undergone therapy. So here, this is firstly illustrated preclinically, but also more recently by the group in Sunnybrook in Canada, that you can also see this in patients. At the Erasmus MC, we then also had the interest of developing the APT-CEST signal. And here we saw the collaboration with Tobias Wood and Gareth Barker from King's College London, together with the iGene study that is being led by Prof. Marion Smits. we here showed that it's really important that if you're going to do APT-CESS, that you image the whole tumor rather than standardly, quite often only looking at a single slice. This means that an example here shown is that the tumor can be quite heterogeneous, and in this patient only, for instance, only approximately 6% of the volume actually showed increased APT signal. And if you take a single slice, for instance here, single slice 4, you might actually miss that this tumor also shows increased amide signal. Further development is ongoing in the item study, and this is done again in collaboration with Tobias Wood by Yulan Wu, who is another one of our PhD candidates, who is further developing, optimizing the acquisition and analysis, making sure that we have the strongest APT-CESS contrast that we can obtain. Here on the right-hand side, he's also make sure that we can assess APT-CESS reliably. So this is a study in healthy volunteers indicating that the coefficient of variation of APT-CESS is very, very low with the technique that we're using. And he's bringing this forward then in the item study again for validation. So here we're aiming to include another 20 patients. Now we're going to look at APT-weighted CESS. We will take biopsies. And here we analyze these samples now for proteomics, just to see how sensitive we actually are with APT-CESS to that protein buildup that we think, that we hypothesize APT-CESS is sensitive to. Here we collaborate with Professor Theo Luider, who is at our neurology department, and together with Leonard Deckert, really pros in doing proteomics analysis. So as I highlighted, we are recruiting 40 patients. That's the aim, 20 in each of these arms. These patients will undergo the same scan protocol. So we will receive a nice data set in the end. Recruitment, however, started last year. So you can imagine it has been quite slow so far, but we do see this actually picking up. However, this also means that at the moment, there are no preliminary results yet to show, other than that we're really aiming to probe the microenvironment further and really use all of these imaging techniques to actually map out the tumor microenvironment in the future. So I'm going to leave you with this cliffhanger here and say to be continued. And I'm looking slightly at the time because I want to spend the last 2 to 4 minutes. Oh, yeah, I have some time. Yeah, perfect. I do want to spend a couple of minutes sort of talking about my passion and so saying how that we should advance physiological imaging, and we can only do that together, in my opinion. So again, bringing this back to the aim I have in the bench to bedside imaging biomarkers, being able to do all this, development, validation, and application, I hope I've already showed you it requires a lot of multidisciplinary collaborations, but certainly will also require collaborations outside your own imaging center and of course outside your own country. And this is why I want to introduce Glioma MRI 2.0 or GliMR. This is a European network that I chair together with Radim Jančálek. He's a neurosurgeon working at the University Hospital in Brno in Czech Republic. And GliMR is a European cost action. That means that we are aiming to build a network in which we connect any relevant stakeholder for glioma imaging diagnostics. These are researchers, clinicians, patients, industries. So we're looking for really bringing everybody together. And for that, we received some Horizon 2020 funding to really build a lasting network. And what we aim to do with GliMR is to advance imaging diagnostics, focusing on being able to do accurate diagnosis, to do optimal patient monitoring, and also optimal treatment response assessment. You can see us here. We got started in September 2019, so this is us in Brussels. More information you can find, of course, on our website, but also in the review paper that got published only last year, in which we introduced the work that we do and how we do it. We have this structured around 5 different working groups that each approach part of collaborating or advancing glioma imaging in their own way. We are advancing MRI biomarkers, our second working group on data sharing and integration, of course, very important in this day and age where safety on data sharing is also very important. Clinical trials is a working group that they focus on the use of imaging in clinical trials. Stakeholder relations, and certainly not to forget is our working group on dissemination, who has done tremendous efforts in reaching out to everyone. basically. We've grown quite a lot over the past year. So what we see now is that we have over 180 members from 30 countries. We're an open network. So again, if you have any inquiries or you're willing to, interested in joining, don't hesitate to get in touch. We organize networking events. So that means that we can organize meetings and training schools, which I have to say, most of the events or activities we organized last year, of course, were virtual. These also included webinars. If travel can happen again, note that we also have short-term scientific missions, so people can apply for a little bit of money to do short lab exchanges to different labs across the world. And we have ITC conference grants. So here we have the coordinators, Ruben and Patricia. Again, more information can be found online about this. I wanted to just finally point your attention to an upcoming event. We are very pleased to be collaborating with MAGMA. So this is the official journal of the European Society for Magnetic Resonance in Medicine and Biology, on a special issue. So here we are focusing on micro to macro scale magnetic resonance imaging of glioma. And I highly encourage any one of you working on this topic to submit your research work by the end of June of this year. So with that, I hope that in the past 20 minutes, I've showed you that in order to advance physiological MRI for glioma, multidisciplinary and international collaborations are really, really key. And I think this lecture series is a very good example of how we can still do that in these difficult times. And if we do that with advanced physiological MRI, this will allow us to probe the microenvironment of the tumor in vivo, where we can really start to address some of the clinically relevant questions we're not answering at the moment. And with that, I really like to thank all my collaborators. I would like to thank the organization once again for inviting me, and thank you for listening. And I will stop sharing my screen.
Perfect.
There we go. Yeah.
Great. Well, thank you both so much, Esther and Zohreh. That was really remarkable. I mean, the amount of spaces that you covered and people's work that you discussed in creating your story and showing your work was remarkable. And thank you for ending, Esther, with sort of that wonderful collaboration, really international collaboration, showing how you're really trying to bring things from the bench to the bedside. Really remarkable work. So I'm going to start. There was a question that came in earlier, but just want to remind individuals who are listening, please put any questions in the chat to our wonderful speakers. The first question was for Zohreh that came in that says GL261 are a good model for GBM as you show. Do you recommend other cell models that can be used in comparison to study the GBM's TME?
This is a very important question, actually, because every one of these mice tumor models in immune competent model have an advantage and a drawback. So collectively, we need multiple mouse models to draw a conclusion to really mimic the TME. For example, the major drawback of GL261 is a high neoantigen load. However, it accurately mimics this T cell sequestration around the periphery as well as the edges in the tumor that we see. There are other models, for example, 005 GSC, that really recapitulates stemness of the tumor, which in GBM is a major question. There is a very therapeutic resistant tumor model with low neoantigen burden, CT2A, that many researchers use, but it kind of grows like a ball, not necessarily like how GBM grows. So again, I emphasize that we need a collection of tumor models. And in our studies, we almost always use at least 3 tumor models, what is available. And when it comes, for example, to GL261, because previous data have shown response to PD-1, we made our own tumor model, which we call it GL261MGH, that does not respond to anti-PD-1 therapy, similar to patients. So these are considerations we have to have in mind before we start doing animal models. It has to be done orthotopically, similar to what happened to the patients. We cannot just do sub-Q tumor. And in that line, I would like to really emphasize that our lab is a pioneer in tumor microenvironment. We offer yearly basis through the Harvard system a course called Tumor course. It has been going on for the past more than 30 years. So if anybody's interested, they can reach out to the Still Lab, ask about the tumor microenvironment course. It's national, international, And that can be a great resource because pretty much we summarize various angles of tumor microenvironment, from immune cells to vessels to various issues and problems we do have in many cancers and many diseases. And I hope I answered your question. If there is anything else, I'll be happy to answer.
Great, thank you. Did you guys have questions for each other? I don't see any unless I'm missing any while I
have one question for Esther.
Mm-hmm.
I think it's super important what you're doing, as I mentioned also before and as we chat. So you're talking about the glioma. How important do you see the impact of this imaging in understanding whether a patient changes from the stage of disease? For example, if a glioma that can turn into a more deadly one. And how do you see that can be done with a minimum potential invasiveness for the patient or along the way of treatment settings?
Yeah, so I think it's a good question, and I don't know whether I have like one answer to that, but I think that, you know, for instance, if you look at APT-CESS, for instance, there is a lot of work showing very recently, even I have to look up the research group again that way. So what they did there is that they scanned APT-CESS just before starting radiotherapy, during giving radiotherapy, and even in follow-up. And we already knew that, you know, at the first moment that you see in the clinic, you see contrast enhancement coming back after treatment, where you're not yet sure whether that's going to be another tumor or whether that's just the effect that radiotherapy has. If you then also do APT-CESS, it really helps you to determine, well, no, actually, this is a patient that is going to progress or that isn't. But even this work showed that even doing that during radiotherapy, 2 weeks into radiotherapy, you can already predict, based on your APT-CESS, which of these patients is going to show progression or is going to show recurrence. And that is really sort of a very good example of saying, well, with advanced imaging and an early moment, you can actually think about, well, this is where I would change my therapy for this particular patient, or here, this is really not working. And maybe sometimes you could decide to, you know, to stop treating or look at it in a very different way. And it's very interesting to see that you can do this with the APT-CESS. Also, some advanced perfusion imaging is also showing that. So it's really about being able to early detect, is there still sort of active tumor present?
Thank you.
Does that make sense?
Yes, absolutely.
I was also interested because wondering whether because you also do a lot of imaging in your lab, right? So do you also do a lot of molecular imaging?
I believe so. I mean, we have 2 dedicated groups who are really doing imaging, both tumor and also the lymphatic system. So Dr. Pader in our lab is heavily specialized in doing lymphatic. And actually, I should say 3 groups. Dr. Fakamura, who is the deputy director of the lab, does a lot of in vivo imaging, as well as there is, and also Dr. Mann, who is also another one of the lab directors, because our lab is a very kind of large lab. It's not, it's a department on its own. So he also specializes in various modes of imaging from devices in vitro and in vivo. So this is heavily done in our laboratory. And we have collaborators clinically who, in collaboration with Dr. Jain, are overseeing the patient imaging, and then we kind of collaborate and publish together.
Yeah, interesting.
I sort of had a question for the both of you. This is like a very new area for me. So learning about how the microenvironment impacts immunotherapy was really interesting. It seems as though it can be relatively small changes, which might be difficult to detect. And I guess this sort of then, my question then sort of moves to Esther in terms of, it's like, how do you validate these measures? Because it seems, and I noticed your samples seem relatively small, again, for someone sort of outside this area of expertise with 20 patients or something. At what point is, when we go from bench to bedside, do you consider one of these scans validated, if you will?
Yeah, no, again, good question. So you can look at, even driving as much as you can forward to decreasing the resolution of these scans. So basically making sure that the scan on the MRI comes from the smallest possible area of tissue. You will always be much, you're still talking about millimeters. And if you do very long structural scans, you can go down to maybe sub-millimeter resolution, but certainly not yet in these advanced techniques. So you are still in a way not comparing at the right scale, basically. So I think the validation is in my opinion, comes from, first of all, being able that you have sensitivity. So for instance, again, taking APT-CESS as an example, that you have sensitivity to at least different amounts of proteins, but you really need sort of the studies indicating, okay, but if you see this type of CESS signal, you see this type of metabolism present. I think it's really about sort of correlating that well, because again, you're never being, it's going to be very difficult to in vivo measure with the same sort of size and resolution that you're going to do anything ex vivo. And I think one of the things that I think is also important to keep in mind is that in the end, I'm driven always in two. I always want to know what is happening, how does it work? If I'm measuring, what am I measuring? To, okay, but what is actually needed in the clinic? And I think not always do you need in a clinical application, you can already use imaging quite far without having sort of the same resolution with your advanced MRI versus what you can do in the lab. So trade-off.
That's an excellent point. Yeah. There's a question that just came in on any thoughts on potential MRI biomarkers for detecting invasive tumor regions that might be growing by vessel co-option and have normal vasculature and hence not visible on T1 post-contrast T2W or VSI imaging?
Yeah, so here, this is a very interesting question as well. And I would then also argue that you would want to look at you want to be looking at trying to see what you can detect or trying to see what is happening and see whether there's an imaging technique that can assess that. Again, for instance, being able to use APT-CESS, or maybe for instance, spectroscopy to look at sort of specific metabolites. Because you basically need one aspect that you can assess that is then different than all the other healthy tissue that you would see around. So indeed, if you see normal vasculature, and for instance, you still see normal oxygen level, then those imaging techniques are not going to give you anything new. However, if you would have normal vasculature, maybe so indeed not that leaky vasculature, but you do actually see already that the vasculature is not sufficient to perfuse the entire region around that vasculature, you can, for instance, already start to detect hypoxia. And that's something that you can then probe. But you would need something along those lines there. I hope that that answers this question.
I mean, I think when it comes to the case of not necessarily human in mice, our lab has published a paper in Nature Biomedical Engineering, which like majorly features the work of Dr. Giorgio Siano, who now has his own lab in France, and Dr. Jane, which they were looking at vessel co-option, specifically a combination of MRI and other, like, methods of imaging in vivo. And they have looked how the tumor cells are crawling and how they are moving and what type of measurement they can do. Yeah. Its application in patient, it will be probably gonna go on in our lab, hopefully in future, but at least in context of mice, that to a level has been done about vascular co-option by tumor cells in GBM.
Yeah, and then I'm sorry, I'm not familiar with this work, but then I would suggest they will use a tracer that binds specifically to any of the processes that you can assess with vessel co-option, I would say.
Great. Well, we're nearing the end of our time here. I want to thank all of our attendees and to have them kind of continue to spread the word. I particularly want to thank our two speakers, Zohreh, our Anne Klibanski Scholar Award winner, and Esther for collaborating with us in this lecture series. Really outstanding talks, fantastic work. And thank you both for doing this today. It was wonderful.
It has been an honor. Thank you very much for the invite and that.
Yeah, thanks a lot, indeed.
Yeah, thank you.
I'm looking forward to collaborating, Zohreh.
Me too. Absolutely. It will be a pleasure. Thank you.
Bye.